Hello, I'm Sophie Rovner from the American Chemical Society. Welcome to this news briefing from the ACS Fall 2020 virtual meeting and expo. We're joined today by Dr. Kimberly Prather from the University of California, San Diego. She's studying how ocean microbes could interact with pollution to influence climate. Dr. Prather.
Thank you. Thank you for attending today. And today, get to the first slide. Today I'm going to talk about research that we've been doing in the NSF Center for Aerosol Impacts on Chemistry of the Environment, or as we refer to it, CASE. And so this is work that's been, I'm the director of CASE. It's been going for about 10 years, and this is the team. So what I'm going to talk about today has the contributions of many, many people from many institutions. It's headquartered at UC San Diego, but you can see that there's a lot of other institutions that are part of CASE as well. And so if you go to the next slide. So the goal in CASE, when we started out, was that I had done field studies. I'm an atmospheric chemist, and we were trying to explain why sometimes you see clouds that are, white versus gray. And, you can actually see over the ocean. Sometimes they're different colors, and that color basically can control the temperature of the planet. The whiter, more reflective clouds can keep things cooler. The darker clouds can keep things warmer. And it was basically hypothesized over 30 years ago that this is due to phytoplankton in the ocean. Basically, our first thought was that the phytoplankton, if you look on the right where there's breaking waves, you can actually see a change in the composition of the seawater. And so what we spent 8 years looking at was how the composition of the seawater changes the composition of the sea spray aerosol. So that's the stuff that comes directly out of the ocean, and how that changes the cloud seeds. So without these aerosols, you would not have clouds. At the center of every cloud drop is an aerosol. So we spent a huge amount of time trying to explain this. We did bloom after bloom after bloom, trying to look at it, and we could not explain the change in the cloud color. So what we've now moved to, and what I'm going to talk about today in my talk this afternoon, and I'll tell you a little bit about here, is these microbes actually also emit a lot of gases. Volatile gases escape the ocean. Those can become oxidized when they hit the air, and they make new particles called secondary marine aerosols. And so that's the focus. And so, 10 years ago, we proposed to NSF, we were going to go and we were going to explain these clouds, but we were going to do it in the lab, and where we could actually control things, and we could isolate the contributions from primary versus these other types of particles. And so we finally have gotten to the point of doing these types of reactions, as shown in the next slide. Which is a study that we did this last summer called.
Sorry, having trouble controlling this.
There we go. You got it? Okay. So this was a study we did last summer. So as I say, we spent 8 years sort of putting the ocean in the lab, and that's shown here in a long wave channel where we use real seawater, we use lights to induce a phytoplankton bloom, and we can basically start looking at now, since we can't explain it just by changes in the primary sea spray, can we change it by looking at these gases and those reactions? And so this is a great topic for an ACS meeting. Now we're looking at reactions that happen to the gases when they get emitted. And so this was a study that involved over 90 participants. It was a massive study. This is usually an empty sort of warehouse with this channel, so you can see all of our visitors. So basically a giant field study in the lab where we, the chemists, can control things. Okay, next slide. So this is the sort of the main result of this study. You can see on top of the wave channel where the waves are breaking. So we have actual breaking waves in this as well, real seawater, real biology. We can actually induce a bloom by adding nutrients. And then these nutrients are like dust, where they feed the phytoplankton. The phytoplankton take off, the bacteria come next, the viruses come next, and you can get this wide range of, they estimate up to 10 to the 15 different molecules in that water. So then we take that headspace, that clean headspace, the air above, and we can actually send it in what's called, bottom left-hand corner is called a PAM OFR. And now we've added the chemistry, finally. We're now going beyond just the biology, beyond the physical production. We're now looking at chemistry, and we can measure the composition of these secondary particles. We can also look at their ability to transform the clouds, and we can ask the question, now can we explain the field observations? And so that's shown up in the top right-hand corner. You measure what's called, you can actually measure directly what's called the hygroscopicity parameter, which has to do with how effectively these aerosols form cloud droplets, convert to cloud droplets. And so you basically something that has a high kappa, high hygroscopicity parameter is very salty, and that's shown on the top. And that's work we did with the primary sea spray. It just doesn't budge. We can do lots and lots of biology. It just stays at about 1. At the bottom, you see what organic compounds, things that don't like water, their hygroscopicity is about 0.2 or lower. Again, that doesn't really describe it either. So in the field, we see this middle range that nobody has been able to explain. As I say, this was 33 years ago, this was proposed. And so when we did this, we turned on the lights, we got the reactions to go finally. We can actually finally, after now it's been 10 years, we can actually explain what is controlling the clouds in marine environments, which is a big deal because these exist over 71% of the Earth. They're referred to often as our planetary thermostat. Aerosols and clouds, that's the largest uncertainty we have in climate. And now we can actually look at what controls the properties of those clouds. And so this shows the team that was involved, Catherine Mayer, John Sawyer, were the leads for this huge project. And we're super excited. And this is the first time we've shown these results. We'll be submitting this paper for publication within the next week or 2. So next slide, please. So I just want to briefly touch on a project that I also mentioned in brief is that, these microbes that we have studied so far are the microbes that are naturally present in seawater. But if you live in a coastal environment like we do, there's also sewage and raw, sort of raw sewage, pollutants. There's a lot of microbes that humans are adding, which is modifying the composition of the seawater and in turn modifying the composition of the air. So we've been looking at the viruses and bacteria that get launched into the air in the surf zone. And we see a lot of viruses and bacteria and basically a lot of things that are present in sewage in the air near San Diego. And so when we had this pandemic start, this is the SARS-CoV-2 that leads to COVID-19, people detected it in sewage. And so we were looking already at the water. And so we decided to take a deeper look at the water in this sort of estuary, which I'll show in a second, look at the ocean water and look in the air. The big question that NSF funded was, do we find SARS-CoV-2 in the water or in the air in the natural environment? And so next slide shows a picture of the region. And so this is well known that there's tens to hundreds of gallons, millions of gallons of sewage dumping into the ocean. It starts in a estuary, it starts in a wastewater treatment plant shown over on the right kind of lower corner. And this starts on the Tijuana side, and then it flows into a river, which flows into an estuary, which flows into the ocean. And so we have a sampling site set up along the coast for air. Also, we collect the water, we collect the estuary, and we collect the river. And so what I'm going to report on today is results that I'll talk about this afternoon, but results where we actually looked in all of these locations to see if we could actually detect the presence of SARS-CoV-2 getting out in the sewage. Because right now there's a massive COVID-19 outbreak in Tijuana. And so the big question is, do we start to see this move into our environment? And if we do, what are the health implications of that? Okay, next slide. So this just shows what the runoff looks like. It goes into the water, and this is 143 million gallons of pollution going in. You can see the brown. So when that gets into the surf and it gets trapped in the surf zone, that can get released into the air. Microbes don't want to be in the ocean, so they sit on the very top surface of the ocean. And so when waves crash, they launch large amounts of whatever's sitting on the surface. So bacteria, viruses, and as I said, we've been looking at that for the natural microbes that are present in the ocean, but now we're starting to look at it thinking of what extra microbes do we see in the air and what are the health implications. And I will talk about the results for the first time. We just got them. They're hot off the press. I'll talk about them this afternoon in my awards lecture. So if you go to the next slide. So this just shows sort of a summary. If you're interested, the award symposium that I'm in is this afternoon between 1 and 3 p.m. I'm the last speaker. I think they're going back to back, so mine will be somewhere between 2 and 2:30, and I will present the results, our very first results that we have in looking for SARS-CoV-2 getting out of the ocean into the air. And I'm going to stop there, and I'm happy to answer any questions.
Are there any questions from viewers? And if so, please state your name and affiliation in the chat window when you type in your question. So first of all, what gave you the idea that ocean microbes and pollution could interact to influence weather and climate?
Basically, it was shown, as I say, years ago that, basically there was something sort of special. The microbes appear to be sort of playing a role in trying to regulate the temperature of the planet. Much like they regulate the temperature of your body, they can actually interact with the air and cool things off, as I mentioned, by changing the temperature of the clouds. And so this was shown a long, long time ago. And I did personally, my group did years of studies out over the ocean. The problem with being out over the ocean is you see everything. You see terrestrial, you see pollution, you see stuff coming out of the ocean. You can never address, what is leading to the change in these cloud properties. And so we basically proposed to NSF, we're going to replicate everything in the lab, the physical production, the waves, the seawater, the biology, and now the reactions, which is a big step forward to actually be able to explain what is it that's changing the cloud properties. And so it's one of those things where it kind of feels like a long time, but in 10 years, we've gotten to the point where we can actually explain the changes in the clouds. So, it was an observation before my career began. And now I feel we're very excited that we can actually see. It's like night and day difference when you start the reactions. So that was the sort of the magic that's leading to the change. It's actually the gases. So one thing I didn't say that I'll say really quickly, other people sort of haven't been able to sort this out in the real world, including ourselves. And a lot of that is because when you go back to the lab, you usually use like really simple systems. And so even if you see something, you can't really explain the complexity of the real world. So having the true chemical complexity in the lab has been key. But the thing that I didn't quite appreciate as much as I do now is that the gases that are coming out, they're really, really reactive. So we are seeing gases come out that people haven't seen before. And that's because when they come out in the real world, they react really fast and they're gone. So unless you just happen to be in the magical spot, you won't see them. So we're seeing them. And then the reason they react is we make them react. And so that's turned out to be really, really a big benefit of CASE.
All right, so that relates to the next question, which is what types of gases and aerosols do bacteria and viruses release?
So the most common gas that the bacteria, or the phytoplankton and the bacteria in combination release is, you hear about DMS, dimethyl sulfide. That is the one that everybody sort of sees over the oceans, has seen it for a very long time. And we also see it in this. I'll talk about this today, but in our study, we see lots of DMS coming out, but we also see lots of other different gases. Essentially the ocean is like a forest underwater. If you think about forests, they emit isoprene and alpha and beta pinene, all these smelly things. And we understand a lot more about the gases that come out of trees than we do the ocean. But the ocean covers so much more of our planet, and we really have just scratched the surface in looking at the gases that are getting out. So it's just classic plants, but they're plants underwater, and those gases are released into the air.
And what types of microbes do you add to your ocean in a lab?
Well, basically we just take what's ever in the seawater that is, present, and we pump the seawater, we bring the seawater, we fill that channel, it's 3,400 gallons of seawater, and then we add nutrients, and then that launches, we take whatever phytoplankton are there, the combination. So a lot of studies have looked at like one particular type of phytoplankton or one type of bacteria. We actually just like happens in the real world. We get all of the biology to take off, and then, so, whatever happens to be present in our local ocean is what we sort of launch to a much higher concentration.
Do you know if there is going to be any efforts to cut off that supply of pollution from Tijuana?
They're working on it. Yeah, this is a really major concern. This has been a problem for decades, and it's just that the infrastructure that's present in their wastewater treatment facility can't handle, and it gets worse. When you see the worst runoff is when there's been rains. Basically, there's just massive amounts of water that go, and I have to get a drink real quick before I choke. Sorry. Anyway, yeah, so there is a lot of work underway. There's actually federal assistance that's come in to help pay to try and make it better. Excuse me. Okay. I haven't coughed all day till right now.
So was it surprising that changing the microbial composition of the ocean in a lab had little effect on the cloud forming potential of the resulting aerosols? Do you have any idea? Sorry. Part two, do you have any ideas as to why this would be?
You know, we are still scratching our head, because if you look at the size of the particles and the composition of the particles, it changed. But yet we couldn't get the ability of them to take up water and make a cloud. It just kind of stayed the same. So before a bloom, when you just have sort of natural seawater, you have these the size that seeds the clouds is about 100 nanometers. Okay. And so those, you know, when you just have natural seawater, they are, you know, largely salty with a little mixture of biological goo, if you will. But over the bloom, they become almost 100% organic, and they still act salty. So we call it this hygroscopicity gap. We and nobody knows why do those organic, you know, that mixture, it's nothing you could ever reproduce in the lab. In the lab, what people had done was they took and they mimicked lipids and things, you know, hydrophobic particles. They made them, and they see a huge shift. So you would expect, based on the organic compounds that are present, sugars, lipids, saccharides, gels, you would expect those to have very different properties than salt. But we just, you know, and so we have not figured that out. Why it seems like it's almost a buffered, you know, Mother Nature has a way. It's almost like a buffered system. And we're still working on trying to understand why, even though we know the composition changes quite a bit, we don't see a change in their ability to form clouds. And we haven't. And we've been doing it for 8 years, and we still can't. So, you know, as I say, now we've moved and we've shifted it, but it took chemistry, took reactions.
So does this result suggest that human modification of ocean microbial community structure might not have a large effect on climate?
No, it says that, to me, it says that it has a lot more to do with the gases that are being released. And if you sort of look at the particle forming potential of those gases, so if they have carbon in them, they're very volatile. They oxidize, they become really sticky, and they make new particles. If you just look at the total amount of carbon that's present in the carbon that comes out of the ocean in the sea spray naturally versus the total carbon that could form from those reactions, the amount that's in the primary cloud particle the sea spray is tiny. It's like 1%, where there's 99% of carbon that, if it reacts, could form. So there's a lot more that when you get these reactions, you can drive a lot more carbon into the particles. And so it kind of makes sense in the end to us now that what seems to control the vast majority of the marine atmosphere's composition are those gases. They're critical. And those gases, we don't know hardly anything about. They come out for all kinds of reasons. They come out of the phytoplankton, they come out of the bacteria, they come out when the bacteria are communicating, they come out when the phytoplankton are stressed. There's like, you know, a huge number, I'd say 10 different ways and reasons the gases get emitted that, you know, we know from sort of soil studies, but we haven't understood for the ocean. And so that's a big push, and that'll be the way we spend the rest of CASE, is trying to understand what controls those gases coming out. So no, what happens, and so that, again, I think, you know, you think about it, billions of years of evolution, those gases have made these clouds with that, you know, 0.7 kappa. Now we throw in human microbes. Now what's going to happen to the gases? Are we going to shift, you know, this from what has been a very relatively stable system? You know, you look over any ocean, you find kappa about 0.7. And as I say, I'll talk about it today, but as things react, they just march right towards that number and they stop. But if you now throw in other microbes, and those start interacting, those start emitting different gases, those start changing things, who knows what's going to happen to the ability to, to get to that sort of cloud forming potential.
And forgive me, I don't think you have addressed this yet, but do you have plans to study the effects of actual air pollutants, such as nitrogen dioxide or carbon monoxide, or a mixture of pollutants?
Yeah, other reactive gases. Yeah, so things like we're looking at N2O5, basically that's a really common one. We also want to look at other radicals. So one of the most abundant radicals, these reactions I should say, I didn't say, were OH radical. But we'll also look at chloride radical. We can look at NO3 radical. I mean, there's a million things we can look at that are, what we really are going to do, I'll tell you, we're developing a new facility where we can actually just bring in outdoor air and react it with the headspace with winds. So it's a massive facility that's going to come online where we can control the ocean temperature. We can make ice. We'll be able to do up to hurricane force winds. So we're going to add winds. We're going to put a smog chamber in the middle. CASE is all about sort of reproducing the full chemical complexity. It is nice. We do go back to the lab and do one gas at a time. But what we're really trying to do is what happens when you just mix it in a controlled way? Can our observations help explain, decades of field observations? That's what we told NSF we were going to do. If we get it back and we can control it, we'll replicate the real world. And we finally just did. It took us 10 years, but we've got a lot more to do. But, we are able to show we can actually reproduce what's out there, and that's been our ultimate goal.
I have a question here from Ryan, who says, I know you touched on this a bit, but has there been a specific effort yet to understand the exact composition of the gas phase emissions?
Yes. So in Seascape, we made measurements of the VOCs through using three different mass spectrometers, because you have to kind of target different classes of compounds. So we had one that was just looking at sort of all VOCs, not quite as sensitive to like the sulfur containing. So we had one that just went after sulfur containing. And then we had another one that was sort of more of a general approach with a different reagent ion. So we have all the information on how those VOCs evolved over the course of this bloom. And I'll tell you, they come out at very different times. DMS is not the only one. Sometimes DMS is not even there and these other ones are taking off. So this is, we're going after this. This is a big, big focus. And we're looking into these other gases. We sort of have grouped them because DMS is can be so dominant. We're grouping the other ones in what we call non-sulfur containing, because sulfur turns out to be really, really abundant. But we also saw, a lot of reduced nitrogen coming out during one of these blooms. We did 3 different blooms over the summer. So we are still unraveling the gases, and we will be for a while.
Can bacteria and viruses in sea spray aerosol be infectious? And has anyone studied that yet?
That's what we're doing. So, basically right now what we're doing, we published a paper in 2018, just saying, do all bacteria and viruses that are sitting on the surface of the ocean, do all of those get airborne when waves crash? And the answer is no. It's a very selective process. Only certain bacteria get out. Only certain viruses get out. And so, now we're trying to sort of understand that. It took us 3 years to be able to reproduce the correct transfer. There's a bubble, you have to get the right bubbles, you have to get the right waves, and now we have it. And when we did that, when people were doing it before, they were just sticking a bunch of bubbles in and everything was getting out. When you actually go to a real wave, it's very, very selective. So the first question we're answering right now is which ones get out? So we have the natural seawater that I showed. Now we're looking for SARS-CoV-2. Does it even get into the air? That's the question that I will answer today in my talk later on, little teaser. And then the next question, which we are not doing. Right now, we're just looking at the RNA and DNA and doing metabolomics. So we're doing genomics, metabolomics of everything that's in the water, everything that's on the surface of the water, and everything that's in the air. And so then the next big question, so we just see the presence or absence of the DNA and RNA. The next question will be, once we find it, which we have found it, in certain places, now we have to collect it in a way that we keep them viable. And that's actually tricky, especially from the air. And we've got infectious disease doctors that are going to take it to the whole next level of, how long do they remain infectious? That's the big question. Just because they're present We hope they're dead, to be honest with you. But we will actually take the next step of the infectious studies as, once we sort of found out where they are. So far, it's just been a survey to see if we see them or not.
All right, thank you so much. The archived version of this session will soon be posted at www.acs.org/fall2020 press conferences.